Hi, my name is Madelaine Gogol. I'm a programmer analyst in the Computational Biology Core facility at Stowers Institute. And today I'm going to be talking to you about spatial transcriptomics. So my talk is Chickens in Space, Our Experiences with Spatial Transcriptomics on the 10x Visium and SlideSeq platforms. Researchers at Stowers study many model and non-model organisms through a variety of techniques. They are really interested in spatial transcriptomics, so they've done some pilot projects using the 10x Visium and SlideSeq technologies. So today I'm going to be talking about that data from the point of view of an analyst working in a core facility. So first I'll talk about the spatial gene expression platforms, the experimental background, and then I'll go through Visium and SlideSeq. I'll compare the two and then give you some take-home messages. So spatial gene expression is any measurement of mRNA with position within a tissue. So there are a number of techniques that have been going on for a while, but 10x Visium and SlideSeq are sort of new and exciting because they take the tissue section, lay it down on a field of barcoded beads so you can see exactly what position each RNA is at. So it's a very high throughput way to do this. So the experiment that we're going to use for this is a slice of a 4.5-day-old chicken trunk. Jason Morrison in the Kalesa lab at Stowers studies neural crest cells and is really interested in the spatial gene expression characteristics that result in proper migration and maintenance of these cells. So this is the experimental system that we're going to use to try all these platforms. So we have single-cell RNA-seq data, Visium data, and SlideSeq data for this experiment. So first, the 10x Visium. It's a slide with 4 different capture areas that are 6.5 millimeters square. There's 5,000 spots per area, and you lay a flash frozen tissue section down on each capture area. This requires a 3-day workflow. To process the Visium data, 10x provides software called Space Ranger, which detects the tissue coordinates, aligns the reads to the genome using STAR, and generates feature spot matrix files that you can read into R using Seurat. By default, Space Ranger only analyzes the area inside of the tissue, so if you want to, you can also use the Loop cell browser to manually select the entire capture area. For us, we did see some signal outside the tissue. So here are the 4 capture areas shown in plots from Seurat, where the number of UMI counts is the color. So the shape that you see, like a crashing spaceship, is the chicken trunk. And then outside of it, we did see some signal. So we looked into that a little bit more, and it ended up being mostly highly expressed genes that were also expressed within the tissue. And looking into it more, we have reason to believe that within the tissue itself, this leakage isn't quite as bad. Analyzing just the spots under tissue with Seurat, we can do dimensionality reduction, we can call clusters. Since our tissue was small, we didn't really have that many spots on the Visium platform that were on our tissue. You can also identify spatial markers using Seurat or another tool called Spark. And now I'm going to talk about the SlideSeq data. This technique was developed by Rodriguez et al. From Broad Institute and Fei Chen and Evan McCosco's labs. The capture area here is a 3 millimeter puck containing 70,000 barcoded beads. You also use a flash frozen tissue section here, and the puck is actually pre-sequenced using solid sequencing in order to get the barcode coordinates that you'll then use to figure out where each RNA was. So this is a 1.5-day lab workflow, and the Broad is planning to open a core to make these sequenced pucks for purchase. So in order to process the raw SlideSeq data, we use SlideSeq tools from the McCosco lab. We have a bit of an older cluster, so it took us a lot of work to actually get it going, and we couldn't actually run it via QSub. So Cynthia Chen from our group met with Jilong Li, the developer, and ended up running the individual scripts in order. So here's some plots from the Seurat package for the SlideSeq data. So we have the natural log of the number of UMI counts per bead on the left. And as you can see, the tissue area is not the only place that is showing a lot of counts. So we do have this leakage or smear outside of the tissue. And you may notice that the counts are kind of low. So it actually gets even worse when you look at the number of counts per gene per bead. So if you're used to single cell data, you're used to sparse data with lots of zeros. Here I'm just taking 20 randomly selected beads and I'm showing you the number of counts per gene. So basically lots of ones, a few twos, and not very many other numbers. So I think there are 50 genes in the whole data set that have a count on any bead greater than 100. So here is the number of features or genes detected per bead. So some beads have 1,000 genes, but many have less than 100 detected on the whole bead. And despite the sparsity of the data, you can still reduce the dimensionality using UMAP. You can still cluster the data, and actually you can pull out things that we would expect to see in the anatomy in the clustering. Since we have single cell data, we can integrate that back to this data set, and that actually helps a lot. So these are 3 clusters we originally identified from the single cell data, and using a Seurat function, which predicts a score for every cluster from the single cell data, we can show where they are localized in the spatial data. Since we did have a lot of leakage outside the tissue, I used this package called gate points to trace around the tissue so that we could kind of isolate that from the background. And there's probably a fancier way to do this eventually. So to compare the platforms, they're really quite different in terms of size. So here's one of the SlideSeq pucks on top of the Visium capture area, and our chicken tissue is shown to scale. So for us at Stowers, where a lot of researchers use model organisms that are smaller, we might like the size of the SlideSeq pucks better. And then the resolution and layout is different as well. So the Visium spots are bigger and further apart, and the SlideSeq spots are closer and smaller. And I'd also point out that you don't get a stained image as part of the SlideSeq protocol. Your mileage may vary, so this is a big table showing some of the aspects of the different data types. But this is, based on chicken and based on Stowers costs, so take it with a grain of salt. This project was a big group project, so a lot of people at Stowers had to help with it, and then people at the Broad were also very helpful, and 10x Genomics technical support. So I'd like to talk about it more and answer any of your questions.